Very good morning to one and all. Mainly today we have Dr. Jaikanth Chandrasekaran, the chief guest for this inaugural function. We, Gokul Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, have organized one-day national level webinar on next generation therapeutics, the convergence of artificial intelligence, regulatory affairs, and endocrine immunology by Department of Pharmacology under the aegis of Internal Quality Assurance Cells (IQAC) of our college. As Gokul Education Society, established by teacher, run by teacher, and led by teacher, was established on 19th February 1918 by Principal T.A. Kulkarni. He was a great teacher and social worker, and the society has completed more than 107 years, having 140 units spread over three zones of Maharashtra, Mumbai, Palghar, and Nasik. More than 125 students are learning at our Gokul Education Society, and mainly this college is named after Honorable late Sir Dr. M.S. Gosavi. He was the Secretary and Director General of Gokul Education Society. He had given his 65 years of life for development of education. And presently, Dr. Mrs. D.P. Deshpande Madam, Secretary, Gokul Education Society, shoulder the responsibility of this college. We do have many colleges, that is senior colleges, uh, art, commerce, science. Then we do also have professional colleges like engineering college, then physiotherapy college, nursing college, polytechnic college. Then first MBA program started in 1968 by our Gokul Education Society. And most of these colleges are accredited and re-accredited by NAC and NBA authority. And among these colleges, we do have our this pharmacy college, that is Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, was established in 2012, and within a short span of 13 years, the college achieved many things. Among that, we do have a 100 intake for BPharm in 2017. Also, DPharm we started at 2017. Then we do have a MPharm program started in 2020 with pharmaceutics 15 intake. 2022 we started pharmaceutical quality assurance with the intake of 15. And in 2022, I am very happy to mention here that our college is also affiliated to Savitribai Phule Pune University as a research center. So we do also have a PhD program in pharmaceutical sciences, and our society do always promote to organize such kind of seminars, workshop, then faculty development programs, international, national seminars, and among that, we have been also promote not only faculty members but students also. Our 7 faculty members visited 7 different countries to attend different international conferences. Few students also attended international conferences, and also society promote do participate in extracurricular activities, and including at Savitribai Phule Pune University level in a different categories of cultural and extracurricular activities. And in the same series, we have been organizing this kind of webinar since the pandemic, Corona. During Corona, ours was the first college to took initiative and organize 7 days FDP, EFDP, and during that Corona period, and near about 1,700 delegates throughout the globe took advantage of that webinar or EFDP, where 18 speakers were there from different parts of the globe: America, England, Dubai, then Sri Lanka, then Nepal, like this. And in the same series, we have been organizing this kind of webinar. Every month, we do organize webinar from 2023. And continuing with this series, today we do have a this national level webinar on next generation therapeutics, the convergence of artificial intelligence, regulatory affairs, and endocrine immunology. Very different thought we do have for this kind of webinar. That is, we have to have asked for a very well-known speaker from academia or industry. Then one of our student, that is alumni of our college, and one of our faculty students. So this is a very different kind of initiative we had taken from last 3 years, and in the same series. Today we do have Dr. Jaikanth Chandrasekaran, Associate Professor, Department of Pharmacology, the Ramachandra Institute of Higher Education and Research, and sir is going to talk on algorithm to therapeutics, our journey with generative AI in drug development. Then second session would be covered by Mrs. Deenal V. Patel. She is a professor, assistant professor at C.V. Patel College of Pharmacy, Mandwa, Surat, Gujarat, and she is going to talk on the immunometabolic role of vitamin D in the pathogenesis of polycystic ovarian syndrome. And then third speaker, who is alumni of our college, that is the prestigious alumni, Mr. Yogendra Patil. He is a senior associated GRAS regulatory operations, Amagan, India, Hyderabad, and he is going to talk on drug regulatory affairs. And I'm very sure all the three speakers would cover in details about the topics, and that could be very much beneficial and helpful for the all delegates. And I'm happy to mention here that today the registration is around more than 1100 for this internal for this national level webinar, and because of the great support by delegates and our management, we do organize this kind of webinar every month, and I'm sure every month we will have a very well-known speakers, and I'm very sure that most of the delegates could again register for this, and our number could be increased, and that's why we are very happy that we are reaching to different parts of the globe, because this is a online webinar, and the registration from throughout the India, not only India but adjacent countries, delegates also have registered for this kind of webinar. So once again, I'm thankful to all the three speakers and all delegates who are attending this today's national level webinar. Once again, I welcome and thank you from on behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research. Thank you. Thank you very much.
On behalf of Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I am Dr. Vijay Lambole, Associate Professor at Department of Pharmacology. Welcomes all the participants to this one-day national level webinar on next-gen therapeutics, the convergence of artificial intelligence, regulatory affairs, and endocrine immunology under the aegis of Internal Quality Assurance Cells, MSG Copper. It's my pleasure to introduce today's first speaker for the session 1, Dr. Jaikanth Chandrasekaran. He is Associate Professor at Department of Pharmacology, Sri Ramachandra Institute of Higher Education and Research, deemed to be University, Porur, Chennai. He is going to talk on the topic today, that is from algorithm to therapeutics, our journey with generative AI in drug development. Regarding his educational qualifications, Dr. Chandrasekaran is honored with PhD in Pharmacology in 2018 from School of Medical Sciences, Jawaharlal Institute of Postgraduate Medical Education and Research from Pondicherry University. He has also taken a fellowship under CSIR SRF, Senior Research Fellowship. He has completed his MPharm in Pharmacology. He has also qualified national level exams, GPAT twice. He has secured first rank in All India Entrance Examination conducted by Pondicherry University for the doctoral program in pharmacology. Regarding Dr. Chandrasekaran's work experience, he is now been working as an associate professor at Faculty of Pharmacy, Sri Ramachandra Institute of Higher Education and Research from October 8, 2022 to present. He has established there the cell culture and molecular pharmacology lab. He was worked as an associate professor at SPTM and MIMS University from 2021 to 2022. The institute, which has been ranked 11th in the NIRF ranking. He was worked as an associate professor at PSG College of Pharmacy from 2018 to 2021. He has established their cancer stem cell laboratory funded by DST FIST. Research areas of Dr. Chandrasekaran are protein kinases, cancer stem cells, phosphoproteomics, artificial intelligence, drug discovery. Regarding his special achievements, he is the principal investigator in ICMR funded project entitled Harnessing of Target Mechanisms for Personalized Therapeutics in TNBC, that is triple negative breast cancer, identifying new biomarkers and combination treatments. He has sanctioned the grant amount of 1.25 crores for the year 2024 to 2027. He has been a recipient of DST FIST funding from 2018 to 2021 for establishing a cancer stem cell laboratory at PSG College of Pharmacy. With this brief introduction, I would like to call upon Dr. Jaikanth Chandrasekaran to start the today's first session. Welcome, sir.
Thank you, sir. Thank you for the warm welcome. And so, shall I? Am I audible, sir?
Yes, yes, you are audible, sir.
Yeah, I'll share my screen.
Please present your screen entire screen.
Sir, present your screen now.
Yeah. Doing it. Is my screen is visible, sir?
Just a minute. Yes, your screen is visible now.
You can control.
Yeah. Sir,
You
is
can
it
start.
moving, sir?
Yes, yes.
Yes, yes.
It's moving, right? Slide one, two,
Yeah,
three.
yeah, moving.
Thank you so much for having me here, sir. No. Thank you so much for having me here, and thank you so much for the principal and then the HOD of pharmacology and Prasad Bengle sir for inviting me to deliver a talk here. So, without further delay, let me move on to the topic, and I'll confess my thanks at the end of the session. Sir, am I audible, right, sir? Is the tone is fine, right?
Going fine. Yes, yes, you are audible, sir.
Yeah. There is no lagging, right?
No lagging.
Yeah, thank you. So, uh, today's talk will be from algorithm to therapeutics, our journey with generative AI in drug development. So, we have developed a small language model and we generate few hit candidates, and I'll walk you through the process so that we have to move along with this technological wave. So, I'll explain you what is first for what is meant by artificial intelligence. So then we'll step slowly into the subject and dwell deeper and try to understand what is what. So artificial intelligence is a biggest umbrella in that we have machine learning, deep learning, and then generative learning. So where this neural network come into play is here the uh, deep learning thing. So machine learning, we have supervised and unsupervised machine learning where we don't have neural networks, and then we need to have a manual intervention to assess. Whereas in deep learning, it has multiple layers. It will try to identify the patterns, and it gives the key nuances about the data. So when we apply deep learning, we can generate new things. That is generative AI. With this brief intro, let me move on to the drug discovery aspects. So these are the drug discovery challenges. These are the vague challenges generally when we speak. So for a drug, this you have heard for many years, many stories about it in your classes. For a drug to come into market, it takes 10 to 15 years to develop a drug, and it costs around 1 to 3 million average cost, and then 90% failure rate in clinical trial. So patient has to wait decades for the clearly explain the drug discovery challenge. This is a broader challenge, broader umbrella challenge. What are the traditional bottlenecks in drug discovery? This gives a meaningful insight why we need generative AI. Not possible. Why? Because we have 10 to the power of 60 possible molecular combinations. That is, our chemical space is huge. So scientists want to test means he can able to test only 1,000 compounds per year. Test in the sense not in silico, in vitro, all these things. So maximum they can go up to near 1,000 compounds per year, and would take longer than ages of universe to explore the total chemical space. Got it. So we have a huge amount of 10 to the power of 60 amount of chemical compounds screening all well. So we are lucky we are in the AI era, and the AI revolution has come up. So what is the next question is whether we can able to design molecules from the scratch. Yes, AI can generate novel compounds with specific properties. I'll show you how we have developed an algorithm and how we have generated the molecule in the coming slides. Like AI for text and art, but for drug molecules, yes, we can also do. So now AI is becoming a master chemist. I'm going to take you through this journey, how AI has become a master chemist. I'll tell you. So, what's our laboratory solution? What we work on, and what's our laboratory solution for designing the molecule from the scratch using generative AI? So, we have a complete AI-powered drug discovery pipeline in our laboratory, so which has been trained on 16 million chemical compounds, and then this model can able to generate thousands of novel candidates. We apply advanced computational filters and validations, and then real results also. I'll show you promising candidate ready for synthesis. We have synthesized few compounds, and we have achieved nanomolar concentration for the few compounds as well. So today's journey will be on AI
Sir, you are not visible, I think.
Fine, right?
Share your screen, sir.
Yes, yes, yes.
And if you lock from your mobile, okay, just mute it because it creates some
Noise.
Yeah,
noise.
yeah, 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 yeah.
Hmm?
Just I
Hmm.
was keeping my phone. I didn't log in. Are you able to see my screen, sir?
Yes, yes.
Yes, sir.
Yeah, yeah. So where I left, I don't know actually.
Okay, okay, no issue.
No
This
issues,
slide.
right? So
You can
I'll
start.
move from
Yes.
here, our laboratory solution. Please interrupt me because I have connected using my mobile network because in our institution they have, live streaming is not permitted. They have made a firewall. That's okay. So we have trained on 16 million chemical compounds, generated thousands of novel compounds. We have applied advanced computational filters and validations. Real results are promising, candidates ready for synthesis. So this is a overview pipeline. Um Interrupt me if the slides are moving. So slides are moving, right? Am I audible, right? Hello, sir.
Yes, yes. Slides are moving and you are audible also.
Yeah, thank you so much. Thank you. Thank you so much. So let me get into the process. Like we have 5 steps. One is data curation and pre-processing. Second is new molecular development. Third comes new molecular generation. Fourth comes the structure-based filters, and fifth comes synthesis and in vitro evaluation for PLK1 inhibitor. So I will break into pieces and make you understand because many audience will be novice, so I'll make you understand about the process. So whatever new terms I'm telling, just make a note and then try to explore. These are the fundamental concept if you really want to do something with generative AI for drug discovery. So the first step is building the foundation model, our massive data set. So now we so now the fundamental question is, how do you teach an AI to understand chemistry? That's the biggest challenge. The same way you teach a child to speak. This is a common analogy through examples, lots and lots of examples. So our AI learns from massive chemical databases. For example, zinc database, which have 16 million synthesized diverse compounds. So we chose that as for pre-training. Make a note what is meant by pre-training. So I'll explain you about these what is meant by pre-training, fine-tuning, epochs, all these new terminologies which you are coming across. So why does scale matter? Why we need to go for pre-training of 16 million compounds? Why does this scale matters a lot? Think about how much human learn language. A child who learn only 100 words will have limited vocabulary, but expose them to millions of words and sentences and conversation, they can develop the fluency. The same is applied to the chemistry as well. Once our aim needs these millions of molecules, their conversation to understand the deepest pattern of chemistry, which atom likely to bond with which makes molecule stable, and how structure relates to biological activity. So we need a massive data set to train the model. For that, we use Zinc database, which has 16 million compounds, purchasable and diverse compounds for our pre-training. So this image explain about how AI learn chemistry like a child learning language. We have to treat them like a child and then make them to understand more and more. So next, I spoke about pre-training. We need to pre-train the model. Then we need to do something called as fine-tuning. So for that, we need a focused set of inhibitors, which is already available. Here, our target is PLK1, that is polo-like kinase 1. So how we get into this target is a different story. If time permits, if they invite for the next time, I'll explain you how to select a target as well. So, but raw data doesn't enough. We need to have focused, high quality examples. From our massive data sets, we identified around 569 PLK1 inhibitors, which is available in Chembel, in PDB. in zinc, all these things. And then PLK1 is why we chose PLK1, that is polo-like kinase 1. It's a well-studied cancer target, point number one. There are existing inhibitors providing validation so that we can fine-tune the model with this existing inhibitor, and there is a clear structural activity relationship and then high therapeutic potential. So it's highly meaningful, and there is no drug in the in market for PLK1 inhibition. There are few companies working at the level of clinical trial in phase 1 and phase 2, and we find this is ideal kinases to work on it. So now let's get into the process, how we started and how we proceeded. So data set preparation and pre-processing. As I said, for our pre-training, we need Zinc 12 database. So it contains 16 million commercially available compounds, and it ensures comprehensive chemical space coverage for drug discovery. So we chose 16 million compounds. After taking 16 million compounds, we need to remove the salts and counter ions to standardize the molecular structure, because they have salts, they have counter ions that need to be removed and make it a clear cut data set. And also we need to eliminate duplicate entries to prevent model bias. Any repetition has to be removed, and we also need to correct any syntactic errors, ensuring data quality is ideal and integral. Okay, so these steps are um, these steps are very much important when you are pre-training the data. So another important aspect is most of them does the drug discovery by taking the canonical smiley and then doing lick prep and then they do docking. That's not an ideal one. So we need to convert the canonical smiley into isomeric smiles, because stereochemical information are crucial for biological activity. We do convert all the 16 million compounds canonical smiles into isomeric smiles, and then we have proceeded forward. Okay, so this is for pre-training. We used Zinc database, and for fine-tuning, we have collated the PLK1 inhibitors from multiple data sets. So multiple database approach for comprehensive coverage. For example, in Chembel, largest bioactivity data set, we identify 821 compounds, and then from PubChem 996 compound, and then BindDB, another one database which have 800 compounds as well, 790 compounds, and from Protein Data Bank, where the proteins are quenched with the inhibitor, we identify 10 compounds, and there is a rigorous pre-processing, as I explained previously, for pre-training. So initially we have, so by altogether we have 2,617 known PLK1 inhibitors. After pre-processing, like removing duplicates, all the steps, removing deduplications, removing counter ions, any syntactic error, we got 569 high quality inhibitors. All right. So there is a 78% reduction ensured only valid non-redundant compound are retained. So the summary here is we have two data set for fine tuning. One is source data, that is Zinc 12 16 million compounds, and then target data set that is curated for PLK1 inhibitors, that is 569 compounds. And now it is ready for machine learning model training and validation. So I was speaking continuously on two terms, that is pre-training and then fine-tuning. So I'll give a small distinction between what is pre-training and fine-tuning. So pre-training means we have to train the model. Okay. After that, we have to fine-tune that model, which will produce the compound of interest for that particular target. So starting point, we'll use random weights. I'll explain you random weights in the coming slide, what it means by weights in the well-known analogy. And then fine-tune will be done on the pre-trained weights. Data size is massive data sets. Here it is 16 million compounds, whereas fine-tuning it is very small, 596 compounds, smaller focused data sets. Cost, very expensive, relatively inexpensive. Time for pre-training, it will take months to weeks to months, and for fine-tuning it will take hours to days. Purpose is very important. Learn general pattern, and for fine tuning, specialized for specific task. Here it is, it has to generate inhibitors closest to PLK1. So knowledge is broad and this is foundational, and in terms of fine tuning, it is narrow and talk specific. So the typical workflow is first pre-train, then fine tune, and then deploy. Where pre-train creates the foundation and fine tune customize it for specific use case. Got it. So how the as I was speaking about data processing, fine tuning, pre-training, deduplications. Is it the biggest task? Not at all. We have developed a Python code. Once you run the code, it will give the pre-processed data within fraction of seconds. So this is a snippet of the code what we have used. At the end, I'll show you a few of the demos on how it works and how fast it will try to convert, and it will give the results all of a sudden. So next we have the data sets, pre-train and fine-tune. Now comes like we need to create a dictionary to teach the AI model. So it's like creating a chemical dictionary that defines every possible word the computer needs to know. So why this matter is just like you need to know H2O means water, the computer need to know means CCO means ethanol. That is CH3CH2O. H is understood. So we will codify as CCO means ethanol at drinking water. So I have given that we have developed a comprehensive dictionary of 113 characters with one vocabulary, and then two character, one character, two character, three character, brackets, branches. So how it has to combine. So this dictionary is very important for the model to learn. So we have developed this dictionary, and then the next is translator. So this is like encoding and decoding. This is like smart translator that can break down chemical formulas into understandable pieces and then convert those pieces into numbers that computer can work with and translate back numbers to the chemical formula. You can see here the input is CCO. It will tokenize. CCO means C stands for 12. It's a random number. It will be assigned by this by our input itself. C means 12. It is fixed, and C means 12. It is fixed. It's a random number, no way related to anything. And O means 14. So number that computer can understand 12, 12, 14. It is encoded. When the computer decodes back, CCO will come back as a chemical formula. That's how we apply this translational way of doing the things. So this is also done in Python and using RTKit. So next we have a dictionary. We have translated to understand the language. Now we need to pack the molecules before machine learning approach. For that, data packaging is important. Packing process, we have to pack in a systematic way. Take a raw chemical data from a text file, use the tokenizer to convert each molecules, package everything into efficient shipment boxes like TF records. TF means tensor flow record. Without TensorFlow or PyTorch, we can't implement any deep learning or Gen AI methods. So it will label each box with molecule length, encode tokens, and molecular ID. So it will try to label this TF record, will try to label molecular length, encodes tokens, and then molecular ID. So a TensorFlow record is very important. We can do without TensorFlow record, right? But doing with TensorFlow record is highly efficient for data processing. It's memory efficient and it's a standardized format. I'll skip this slide and move to the next so that you can able to understand. Real world performance examples are imagine you are training 1 million compounds without TF record. Reading a text file will take 30 seconds per epoch. Epoch means I'll explain you what is epoch in the coming slide. And then memory usage is greater, 8 GB. Format tracing recurs every time. So with TF record, 3 seconds per epoch, memory usage is 2 GB, and formatting tracing. Once done, it can be used during creation. So complete workflow till what we have seen is the input is the compound CCO. This is the zinc ID, ethanol with an ID. Now we will give this to the SMILE vocabulary, which provides vocabulary that I have shown you the dictionary. That CCO, it is coded as encoded as 12, 12, and 14. Now it is fed into 10th floor record. So it will lens how many characters are there. It is 3, so it will mention as 3, and then it codes the molecular ID and the name. So that's how all the compounds will be packed, and this will be loaded and batches data for training and fed to neural network of 32 molecules at a time, and then we process all this aspect. So this is a workflow example. Since we have the data, 10th floor record is ready, we need to move towards subjecting to the neural network. So we have different neural network. We have used GRU. So for that, I'll make you understand what are the different neural networks, their key strengths in this slide. The first basic model is RNN. It's a sequential data, and the data type is time series and text. Uh, Key strength is can handle variable length sequences, but the limitation is the vanishing gradient, the memory. Memory is the biggest problem. Short-term memory only. It can't remember the long term. Next comes the CNN. CNN is for spatial data. It's for imaging and 2D patterns. So anybody is working on the computer vision, image processing from the X-ray, NMR, whatever data, any images, we should use this architecture CNN. And then next model is the LSTM, long short-term memory. It can handle the long sequence, text, speech, and time series. The memory is it can able to memorize, remember long and short term. Whereas in GRU, it's a simplified version of LSTM because it consumes a lot of computational power. So it can also handle the sequential data. Since our SMILE is a sequential data, we use GRU, and it's similar than LSTM. It has memory gate and reset gate. It's less expensive than LSTM. It also have the same long-term plus short-term memory. And the next one is the transformer. This is how your chatbot, ChatGPT, all works using this transformer model. So we have built the transformer model as well, the small scale transformer model. I didn't show the transformer model for this presentation. I'll be showing about our GRU model for our presentation. It's the fastest alternative to LSTM, but transformer model is the state of art for NLP and large scale tasks. Since our use case is designing a drug, is more than efficient than going to the transform. So this is our GRU. The
Sir, are you there?
Yeah. I think there is an error with your
Please share your screen once again.
yes.
Screen is shared. Screen is shared. So you can continue.
Is it
Yes, you
visible,
can
sir?
continue, sir. Yes,
It's
you
visible,
can
right?
continue, sir.
Yes. This is a GRU architecture. 113 tokens will be converted into a 256 dimensional vector. Little bit of mathematics, no need to worry. Parameters, 29,000 parameters it will do. So what is the purpose? It will convert chemical tokens into dense representation. Right. Um, Dense representation. Then this is a GRE core, that is the neurons. We use 512 neurons. So this 256 vectors are fed into 512 neurons, that is neural networks. So then what it does is it will parameterize into 1.6 million parameters, and then sequential memory with chemical context. It will try to memorize the sequential memory with chemical context, and then there is a dense output layer from this 512 dimension. It will try to predict what will be the next smile token. That's the model architecture. With this model architecture, I'm moving into the real scenario, how the training happens. So we need to train. We have a Zinc database, which is converted into TensorFlow record. So the batch size is 32, and then epochs. What is epochs? I'll explain to you in the coming slide. So these are the standard parameters generally we use for machine learning approach. I'll get into the next slides. So the question is, what is epoch? Imagine you are studying, it's a study session. An epoch is like one complete study session where a student goes through the entire textbook once from cover to cover. So we have 16 million compounds. So if I train a model for one epoch, it will try to learn all the 16 million compounds at once. So one epoch complete pass through all the training data, the real world learning analogy, right? Got it. So for example, you make our machine to read all 1 million. Here it is 16 million molecules once. So once it learns, it will learn little bit from each one. By the end, you will see every molecules exactly once in one epoch. When we do it for multiple epochs, epoch 1, first time reading for your understanding, you understand basic pattern. When you read it again, that same textbook, you notice more complex relationship. In the third time reading, you start recognizing advanced pattern. And when you read the same textbook for the 10th time, you become an expert to predict molecular structure. The same analogy is applied for pre-training as well, right? So the training loss has to come down gradually. I'll explain you what is training loss and what is validation loss in the coming slide, right? This is a training, as I said, pre-training and fine-tuning. This is a training loss and validation loss. So what is training and validation loss? So what is training loss? What is it? How well the model performs on data it already learned from. So analogy, you perform on a practical problem you have already studied. It is expected it should decrease as the training progress. At one epoch, the training loss is 4, at 10th it is 2, at 20 it is 1, and 30 it is 0.5. So the learning is happening in a very good and in a healthy way. Right. So we need to also understand the validation loss. So these are the data which we have already shown, and then so it is performing well. If you show the model the data which it has not seen before, and how it is performing really makes sense. How well the model perform on data it has never seen before. Okay, analogy, you perform on a surprise quiz with new problems. So the test, if you truly understand the concept, or it just memorizing the answer. So that's the purpose, whether it is truly understand the concept and it is trying to solve, or it is just memorizing it. So that can be done using the validation loss. If you give a validation state, so here you can see 4.2, at the 10th epoch it is coming down 2.1, and at 20th epoch it is 1.1, and 30th epoch it is 0.6. The gap is also good, healthy. So when we need to stop, we can run these epochs continuously, 100, 200,000, not required. When we need to stop? When there is overfitting, when the model try to memorize, you can see here. So at epoch 1, training loss is 4 and validation loss is 4.2, and 10 it is gradually coming down. This is also coming down, and 20 also it is coming down. Whereas in 30, this is coming down, this is going up when compared to 20th epoch. So the gap is growing, and this is the area of overfitting. So we need to snapshot which is best. So what is the best weight is the student best performance, where the training loss and the validation loss are less. The best weights are likely saving a snapshot of your student's brain at the moment they perform their absolute best on the surprise quiz. So we need to focus more on the validation loss. So this is, for example, in this case, 20th epoch is the best weight. That weight we need to take. On that weight, we need to fine tune our model. So pre-training is fine, and now we are moving to the fine tuning. So we have for pre-training we have captured the best weight. Now we are moving forward to the fine tuning. As I said, fine tuning should be done on PLK1 specific molecule. It's domain specific. So load the pre-trained weight that we have already identified in the pre-trained data set, and then apply the same training transfer learning for specific chemistry. Here you can see the training loss and validation loss. You can see here the training loss is also coming down gradually at 16th epoch, and validation loss is also coming down gradually. At 17th epoch, the validation loss is climbing up, and also in 18th epoch, it is also greater than the 16th. So this is the snapshot. So I have to capture this 16th epoch. On that epoch only, I have to generate the molecules. I hope you can try to understand a little bit, but if you have any questions, or we can discuss at the end. Next comes, we have the best weights. Now we need to generate sample out of it. So the challenge is we have to create diverse and valid molecules. Our AI model gives. So we have applied three strategies. One is greedy, top K and top V, and hybrid sampling. Greedy means I always try to pick the most probabilistic token, like always choosing one first search from the Google, what is most probable, safe and predictable molecule. So it will keep on repeating. So in greedy, we'll get only one molecule. Whereas top K means it will pick random from top 50 options, choosing from 50, for example, for your analogy, choosing from top 50 Google search, and it will try to convert to combination. And top V means pick from option that add up to 90% likelihood, choosing until you are 90% confident. And it's a smart variety based on the context. And hybrid, which implies both K and P, and then it will try to generate. Like that, we have generated 10,000 in top K, top P, and hybrid 30,000 molecules. So now we had around 30,000 molecules. Next is how many molecules are unique scaffold. Unique scaffold in the sense which is not present in already existing PLK1 inhibitors, which are really unique. The total number of unique scaffolds in 10,000 or 6,851, and top K is 8,022, and hybrid is 6,855. And then next important aspect is Lipinski rule of violation. How our model is performing well or good. So 70% of the compound have zero violation. Yes, our model is doing well. And 19.95% has one violation. So 90% of the molecule can be taken forward for the drug discovery. So when we do deduplication, because this generative AI, this GRE model can also produce the same molecule, we need to do deduplication. So from 27,000 out of 30,000 molecules, we had 27,152 molecules for the next level. So this much we have covered as of now. Now next moment is whether the learning has actually happened. So for that, what we did is there is something called as uniform manifold approximation. It starts from principal component analysis, then moved forward to TSNE. Now it's UMAP, and then it is TMAP. What it does is it will try to capture both the local and global association in a two-dimensional space. This is uniform manifold approximatory projection. So what we did is we need to know whether actually our model learned and the transfer learning has happened or not. For that, what we did is 3,000 molecules from the zinc database we randomly chose. Got it. And then this is called a source. These 3,000 molecules are source, and all available PLK1 inhibitors, that is the target. The green color are the target, and 3,000 generated molecules from our hybrid sampling and other things. So our generated 3,000 molecules. So you can see that the transfer learning has happened excellently because the source data is here, that is zinc molecule is here, and all our generated molecules are either near the PLK1 or away from the zinc. So the transfer learning has happened well and good. So our model is working fine. So moving on to the phase 1 is completed. So we need to calculate the molecular weight, log P, everything is computed in the code itself and it will come. I'll go to the next step. Next is the phase 2, that is advanced chem informatics. As of now, phase 1, we have generated molecules which are relatively novel. Now we need to adapt advanced chem informatics, which generally almost in all academia and then industry they do, but they don't know advanced chem informatics filters, what they are doing and applying. Right. So first and foremost, ligand preparation and strategic filtration. So we have 27,153 ligands. So we did all in Schrodinger and then other online and version of the softwares. And then AI generated molecules, 27,153 molecules have been converted into isomeric smile. And then we process for ligand preparation. This yielded 55,096 compounds. Then comes the application of filters, which generally most of the students, even faculties, and most of the people don't apply. First is tautomer filter, size filter, solubility filter, drug likeliness filter, synthesis filter, and finally this will leads into high quality leads. That is, here is 25,217 from 55,096. I'll quickly move on to the slide and show you why and what. What are the filters we have applied? Tautomer filters probability should be greater than or equal to 0.8. Why? Because it is the most stable form of the molecules. So we need to select only the stable forms. Because molecule can exist in different form, we need only stable form. Next is ionization penalty. Should be less than or equal to 2.0. Does this molecule behave predictably in the biological fluid? Why need? Drug need to maintain their property in both blood and stomach acid. Molecular weight ideal 150 to 500. It should be in right size. And then log P 0 to 5. Water and fat and water balance. So can this molecule cross the cell membrane? Because drug must be water soluble while absorption and fat soluble when it is entering into the cell. And the QED should be greater than or equal to 0.5. This is a drug likeliness score. This filter should also be applied. And then synthesis feasibility. Whatever molecule you prepare, if it is impossible to synthesize, then it is of no use. So we need to apply this filter and then Lipinski rule of violation, whether this molecule can be given orally or not. We kept it can violate only one. Okay. So now we have found out a valid drug-like molecule by applying all these filters. The next new filter is, will this drug actually work when it reaches the target or not, is the next question. Okay, so for that we have generated Pharmaco4 model building. This is also Python coded. I'll show you at the end of the session. Just a simple code will give you all the data and structural based drug design by insight into PLK1 inhibitor. So I'll skip this slide and move on to it. So once we apply this, it will go and pull. It will go and retrieve all the PLK1 inhibitors along and its crystalline structure from PDB, and it will try to map aromatic phase to H, aromatic phase to phase, hydrogen bond acceptor, hydrogen bond donors, hydrophobic, and ionic negative. So it will try to produce a heat map, and it will say, for example, here 133 in X-ray, residue 133, 15, and 22. It's 15 times it is existing in all the PDB structures. So from the heat map itself, we can try to identify the hotspot residue, and also we can able to from this we can identify what are gatekeepers and what are the hinge region, whether your kinase is in active form, whether DFG is in or out. All these things can be captured using this heat map itself. So this also coded. So why these regions matters a lot. Mere talking is not going to suffice anything. We need to understand the active sites more and more detail. Hinge region, for example, in our PLK1, the hinge regions lies between 132 and 134 amino acid. They have key interaction like H bond and hydrophobic interaction. So our molecule, when I am designing, it should have the property of forming H bond, and it should also have hydrophobic. So why it's very important? For anchoring, affinity, it mimics ATP, and classical kinase hinge binding. So hinge region is very important for the activity. Next comes the gatekeeper. So gatekeeper is very important because it's selective. It determines the selectivity. It blocks the activation loop and engages subtype specific sites. So this is also very important. Solvent front and back is not really important for us because we are designing a competitive ATP inhibitor. For if I am planning for allosteric target, then this is very important. Whereas an aromatic sites, these are existing in the hinge region as well as other regions, so it will stabilize the binding and can enhance affinity and specificity. So a lot of critical nuances and critical things has to be taken care when we look into the pharmacophore. So based on these insights, we have selected the features. So 2 acceptors, 3 acceptors, 1 donors, and then aromatic ring. So this is the pharmacophore e-pharmacophore model. And before proceeding into docking, we redocked and confirmed our algorithm is working fine because the RMSE is 0.679, the acceptable is less than 1, and it's fine. It's very less. And this is the pharmacophore. Now we will subject all the filtered, liquid filtered, and applied filter ligands into this pharmacophore model. So that is called as phase screening. Here also we need to apply a lot of filters, and you should know why these filters make sense. So phase score is the overall fitness score. The score is greater than 2, it is excellent fit, moderate it's 1.5, and poor 1 less than 1.5, we will leave that molecules. Then it comes the align score. Does the drug 3D shape matches the target pocket? For example, if the target pocket is L shape, and your drug should also be L-shaped, then only it can go on bind. That will be given by the align score. This makes a lot of sense in drug discovery. Then vector score, directional interaction. So if a protein has a positive charge in one spot, drug should have negative charge pointing there. This is also make a lot of sense. So vector scores, align score, and then volume score. Whether the drug is too small or bulky, it makes sense. So for that, we have applied filters like phase score should be greater than or equal to 2, align score greater than or equal to 0.1, the optimal threshold is 1. So these scores are applied, and then we got 324 highly preferred and 132 moderately preferred molecules. So we got 324 highly preferred molecules, and that enter into the docking stage. So the key lock and key testing laboratory model is so it actually tests them in the real log to see how well they work actually. So this is a very critical step. So far, as a basic filter, is it a valid key that what we have did, and then in pharmacophore based filtration, that is space filter, does this key look like it should fit? In docking, does this key actually work in the log? So we are here. I'll also explain you about this molecular docking and why all these scores matter sense. We always look into docking score, that is So observed way of doing docking, but we need to also look at some other key parameters how industry operates. So docking is a sophisticated computational simulation that takes your drug molecules and then target, then physical testing, how they interact in 3D space. That's the definition. Now let's get into docking score type. We have docking score, we have glide E model, we have ligand efficacy, we have glide energy and glide Z score. So these three are very important when we are doing docking. So we can keep the threshold accordingly. You can keep the binding strength of the drug. Force need to pull the key from the lock. That is analogy what it measures. So it will primarily measure the binding affinity. These are the threshold we have kept. And then glide E model. This is very important. It will give overall thermodynamic favorability. Reflects stability of the bound complex. If your docking score is high and then E model is relatively less, then you have to throw away the molecules. Both need to be high. That's the crux. So because it will reflect the stability of the bound complex. Next comes the ligand efficacy. So binding strength per heavy atom. So if the heavy atom is less and the binding strength is good, then it will always reward the smaller efficient molecule than the larger bigger size, because they have a lot of heavy atoms and bang for bulk efficacy per atom. So these filters will try to overcome the false positive results. Got it. So by applying all these things we have achieved from our compounds, we have docking score. You can see the docking score is minus line, and glide E model is fantastic, that is greater than 100. You can see almost all our top hits, and the ligand efficacy is also good, and heavy atom counts is in the acceptable range between 25 to 35, and excellent and borderline. So next comes the important question. So whatever molecules, our ultimate aim is to have a molecule which is novel and it can be taken forward for IT. That's an ultimatum. So what we did is then we move forward to SciFinder and SureChemical to find out the molecules which we got it, how much is novel, or number of substructures are available, or the scaffold similar to known PLK1 inhibitor, do they have or not. So 6 molecules are novel, but they have scaffold similarity to PLK1 inhibitors. So even though they have the activity, since they have the similarity, we may have IP infringement. So we skipped all those molecules even though they show a promising activity. Next is 9 molecules are novel in both aspects. It's novel, no similar substructure, there is no scaffold similarity. From that 9 molecules, we chose 5 molecules for synthesis, and 3 molecules are showing at the nanomolar activity. I didn't show the in vitro data. So this is docking score results of 5 top 5 hits of molecules. So these are the key interactions of the molecules. You can see pi stacking, you can see this is 133, and then in gatekeeper region, so all are retimed. And this is a comprehensive pipeline I initially shown you, right? So from pre-training, 16 million compounds, fine-tuning, these many databases we use. For fine-tuning, we kept the compounds which have IC50 less than 1 micromolar, and then we put them in our GRE model architecture, generative AI. The prerequisites are here. Then we did epochs, the best weights are taken, then the samples are generated, then we validated whether the transfer learning happened or not, then the feature selection using pharmacophore mapping, and then different types of filters, and finally IP, novelty and IP assessment with synthesis feasibility. So now we have synthesized the molecules and we have checked their in vitro activity, and it's showing it at a nanomolar. So our model has successfully implemented, and this same strategy is applied for many kinase inhibitors which we are doing in our pipeline. So this is my team. 2 are missing actually. I have a grant of 1.25 crores from ICMR. So team member is she's Dr. Manasa. She's the postdoc researcher working in my ICMR project. She's leading the project, and then Megal. She did her PG from MS Leeds UK, and she is working as a project technical associate. And Mr. Rajesh, who is a full-time PhD scholar who work on plant-based anti-cancer lead use in metabolomics. Dhanushya works on cheminformatics and machine learning-driven kinase drug targets. And another scholar, Preeti, works on exploring novel checkpoint inhibitors from cancer therapy. And Karan works on QSP modeling in area of DNA damage response and combination therapy. And Nikki works on CCN amplified ovarian cancer for target kinase vulnerability. As of now, we have one PhD position currently open in our laboratory, and this is our laboratory. And this is a small snippet of our laboratory. We have all basic equipments: qPCR, PCR, Western blot, ELISA, CO2 incubator, fluorescent microscopy. All these things are there. And if you need more details, you can contact me through my number, or you can email me through any of the email. And that's all from my end. I know it's very difficult to make you understand, but with a less amount of time, I make you understand that these can be done using generative AI. So the key terms you need to remember are pre-training, fine-tuning, epochs or epochs, then sampling strategy, filters which I have shown. If you apply all these things, this is very easy to. And thank you so much. And I'm happy to take questions if it is any. Thank you so much. I hope I have completed in on time. I think so.
Dear participants, do you have any questions? Please write your questions in the chat box. As such, I have not seeing any questions till time. Okay, there are no questions, Chandrasekaran sir. So,
Thank you, sir.
thank you so much. Yes, thank you so much, Dr. Jaikanth Chandrasekaran sir. Uh, he has given a very insightful thoughts on the algorithm to therapeutics with the help of artificial intelligence. He has covered many key points in his session, like complete AI-powered drug discovery, PLK1 protein inhibitors, neural network architectures, and most important is the docking analysis. So thank you so much for giving us the thoughtful knowledge on the generative medicine, sir.
Thank you, sir. Thank you so much.
Uh,
Thank you, principal, and thank you, the managing personalities, HOD of Pharmacology Department. Great initiative conducting all this program from Pharmacology. As we are Pharmacology Department, I'm happy to see this type of initiative. Thank you so much for having me. Thank you, sir.
yes, you are welcome, sir. Sir, please accept this certificate of appreciation from our
Thank
side.
you. Thank you.
Thank you once again, sir. Uh, you can leave the session right now, sir.
Yeah, thank you, sir.
Thank you so much.
Thank you so much.